A big hello to all my PU2 aspirants. I know that the exam season is here, but the exam fear should not be here because we are there for you, my dear children. And you all are very, very hardworking. So why hardworking people have to fear about anything? You are going to ace your examination, and to help you, we are here, right? So today we are going to discuss about the PU2 most important diagrams, and it will be in one shot. So you don't have to lurk around anywhere else. You will be getting all the important diagrams in one video. So I saw a cute comment, that is by Dicot, saying that, hello ma'am, please do second PUC all diagram videos, please ma'am. So this was under the 10-day strategy video, which also is a very important video. If you have not watched it, you should definitely watch it. So yes, your wish is considered. I am going to make it, and it's not just her wish. I know a lot of you have asked in other videos as well. I just want to show you one thing. It's not like I'm showing you a comment or something like that. It's just that we consider what you are asking us, and we plan our day accordingly. So do not worry about anything. Please keep asking whatever you need. We will be here to provide you that. Okay, now my dear children, for boards biology, this is the schedule. So Wednesday you have model paper 1, Thursday you have biology all markers. From Saturday you have model paper 2 solutions, Saturday you have biology all 2 plus 3 markers, as well as all diagrams, that is this. And February 24th, that is Monday, you would be having all the important 5-marker question, and February 26th you would be having the passing package. So please do not worry about anything. We consider you a lot and we have planned everything accordingly, but still if you need something else, you have to tell us. We are here for you. Now let's start without wasting any bit of your time. So principles of inheritance and variation, my dear children, over here if you go on page number 54, there are 7 pairs of contrasting traits. You should be knowing all the pairs really well. Okay, the name, everything, seed shape, then seed color, then flower color, pod shape, pod color, flower position, and stem. The stem is the only thing that is kind of different. Rest everything has everything in pairs. So seed shape, seed color, flower position, pod shape, pod color, and then you have stem height. These are very important. Now apart from that, if you see pedigree chart, my dear children, which is mentioned in pedigree analysis on page number 72, they would be giving you this diagram and asking you what does this mean. So usually they give this and this. So this is when mating is happening between relatives, consanguineous mating. Okay, now this is normal mating that is being shown. Now when it is colored, that means it is affected individuals that they are trying to show. Now I have included all the diagrams from which you can get questions as well. Not to represent diagrammatically, but you can get questions as well, so that I cover all the things. Now over here, my dear children, on page number 73, a pedigree analysis again. So you should be knowing how do you understand a dominant disease or recessive disease. So dominant disease would be there in all the generation. So if you see here in parental generation there is 1, and now when they fused in this generation also you will be finding 2. Now when they fuse you will be finding in the next generation as well. So dominant traits can be identified like that. Recessive ones, they can skip generation. They would be skipping generation, and in this you would be noticing that the parents may not show it, but the children would have it. The parents are not showing it, but the children are having it. So this is an example of autosomal dominant trait, that is myotonic dystrophy, my dear children, which is just given under the picture. So this example is really important. Now over here, autosomal recessive trait, you have sickle cell anemia. Cool. Now next, talking about molecular basis of inheritance, you have the structure of nucleosome on page number 83. This has been used as to, you have to draw it. So please draw it properly, make the nucleosome, that is histone proteins, and then the DNA wrapped around it. This whole structure is known as nucleosome. Then you have histone 1 protein also. Okay, and now over here, on page number 92, you have transcription unit. Again, a very famous question. You should always remember this is a template strand and this is a coding strand. So on the 5th 5' end of the coding strand, you will be having the promoter region, and the 3' end of the coding strand, you will be having the terminator region. So if at all you forget things, just remember this is 3, because transcription happens in 5' to 3' direction. So obviously it will be coming and binding over here and making a strand like this. Okay, so promoter, terminator, and the structural gene which is there, template strand and the coding strand. This is a transcription unit. You might be given the template strand, you would be asked to make the whole transcription unit with this. So what do? You make the complementary base pairs over here. Make sure the 5' is towards down and 3 is up. Okay, while making so. Cool. And if you're asked to make the mRNA, what do? You can, whatever you code you wrote over here, that will be there in the mRNA, but it's an RNA. This is DNA, right? So in RNA, you replace all the T with U. Cool. Now, there is a structure of translation. Here again, question can come, that is, what is the anticodon for valine? So, this is the anticodon for valine that is there on the tRNA. What is the codon for valine? That is GUU, my dear children. Now, there can be a question asked, that is, what is there for alanine? So, in that way, similar way, you should be knowing it. Okay. Now, for stop codon, if there is a stop codon, there won't be any tRNA coming and joining as well. Again, look for the page number, it is important. Now, very important thing, lac operon on page number 101, you would have to explain it, you would have to draw it and everything. So, please know you have every structural gene has a promoter region. So, this regulatory gene also have a promoter region. Similarly, the structural gene over here also have a promoter region. They also have an operator region. On this operator region comes and binds a repressor protein which does not allow transcription to happen. You should know what does Z do. Z produces beta-galactosidase, Y produces permease, and A produces trans-acetylase. Okay, and beta-galactosidase is there for breaking down the lactose. Okay, permease is for letting in the lactose into the E. coli and things like that. And lactose and allolactose are the inducers which would come and bind on the repressor, such that the repressor is not free to bind over here. So, if it is not coming and binding, that means transcription can easily happen. So, lac operon, very, very important. Now, again, you have tRNA structure. So, please remember, it's on the 3' end of the tRNA that the amino acid come and attach. You call this process as amino acylation or charging of tRNA, and it needs a lot of ATP. Needs lot of ATP. Now, the shape of the tRNA is considered to be what, say, clover shape, but actual sense it is what, inverted L. It is inverted L. Okay, again, structure is important. Now, human health and diseases, this is important, that is the stages of Plasmodium. Go through it, you should be knowing, because the Plasmodium whole cycle is important, so you can get question from any stage. Okay, and here also the picture can be given and you might be asked, what is this? So, this is thymus, my dear children. Thymus is a primary lymphoid organs. What are these? These are lymph nodes, they are secondary lymphoid organs. These are lymphatic vessels. So, please go and study thoroughly about the lymphoid organs as well. Now, that is an important question for 3 marker as well as what, 5 marker as well. Okay. So, that is on 138 page number. Here all the stages, that is, where does it come and affect first? Plasmodium, it will affect your liver first, and then it will go affect your RBC. In the RBC, they form gametocytes. Okay. So, all the small information. Do they form gametes in human body? They do not form gametes in the human body. They make the gametes in the mosquito's gut. Okay. Please remember that, and they fuse to form the sporozoites. The sporozoites is something that is given back to us and the stage goes on. So, please go through it completely. Now, human health and diseases, structure of an antibody, most important diagram every time it comes. The two heavy chains, 2 light chains, variable region, constant region, variable region, constant region. The disulfide bonds between this and the antigen binding site. Again, they can ask you schematic representation of the replication of retrovirus in HIV. So, all this diagram is important. The HIV virus coming, utilizing our machinery, making their copies, what enzymes are being used, reverse transcriptase enzyme is being used. All this information are really crucial for you to know. Okay, so please go and draw it. If you have not seen the Brahmastra series, we can watch it also in there. At least for the chapters which are high weightage chapters, no, you can definitely go through it once. Now, human health and diseases, this structure of morphine given on page number 142, my dear children. You can be asked, where do you get this from? From which plant? It is from what? Papaver somniferum, opium poppy. Now, this structure is of cannabinoid. So, always remember cannabinoid. Whenever someone has cannabinoid, it is like, actually cannabinoid is not associated with hallucination in your book. But if you have cannabinoid, you get hallucination. So, you do not know what the steps are there or not. You can think like that, or in whatever way you want to remember, you can remember. I will give you an easy way to remember this at least. So, it looks like a O. Right, it looks like a O. So, you get it from opium poppy, that is Papaver somniferum. Now, this datura is given on 143. It is a hallucinogen, and then this is the opium poppy plant, my dear children, from where you get this. Now, the latex of the opium poppy, from here the milk shapes things, milk sort of thing, latex of the poppy comes, from which you get morphine as well as heroin, that is drugs. That are the drugs. Okay, so please know how to identify it. So, whenever you have a flower like this is datura. Whenever you have a pod shaped thing, that is what? Opium poppy. Now, sexual reproduction in flowering plants, my dear children. Again, very important diagram, that is transverse section of anther. You should be knowing all the layers, epidermis, then endothecium, then middle layer, then microspore, mother cell, then tapetum, then sporogenous tissue which is there. So please know that. Apart from that, you have the pollen grain structure. They can ask you, is vegetative cell floating in the generative cell or generative cell floating in vegetative? So it is actually generative cell that floats in the vegetative cell. They can ask you for the structure of the nucleus. So in vegetative cell, the structure is irregular nucleus. In here, it is spindle shaped nucleus. Okay, and what is the use of vegetative cell? It provides nourishment, nutrients. Now, the structure of megasporangium is being asked. Now, that is an inverted thing that is being put over here, but you should know the actual structure. So there would be ovule. Okay, ovule is covered by two integuments. Where the egg is there, you call it micropylar end, and where the other end, where the antipodal cells are there, you call it the chalazal end. Okay, and then you have the placenta in the ovarian cavity. So it looks something like this. I'll just make it and show it to you if at all you're confused. Suppose this is the ovary. So over here you have the placenta. The placenta is attached to the ovule like this with the help of some structure that is known as funicle. Now this ovule has two integuments. One is this, and there is one more, that is this, that is outer integument. Okay, and then here there will be embryo sac and nucellus and all these things. So you should be knowing about this body. This end is known as micropylar end, where there is an opening, and this end is known as chalazal end. Cool? Now here again, important for 1 mark question. You have, you might be asked what is syncarpus, apocarpus, examples you might be asked. So this is an example of syncarpus, as in you have so many carpels, but they are together. So syncarpillary condition is being seen in papaver, my dear children, and apocarpus, as in there are so many carpels, but they are in different zones. Okay, they are not together. So that is apocarpus, and that is seen in Michelia. So the examples are given up over here. So these pictures are also equally important. So some people, what do is that they just tell you what is important to draw. So this is not just an important thing to draw, you should be able to identify also properly, right? Which is why I am putting it over here. Okay, now this is an anatropous ovule, as an ulta ovule. Chalo. So over here, usually what happens is that you have the funicle and placenta towards down, but here in anatropous ovule, the funicle and the placenta will be towards upside. Now, again, so many pictures up here which you have to remember, my dear children. Mature embryo sac, obviously. Chalazal end is where you have antipodal cells, and the micropylar end, as I said, egg apparatus is there. The egg and the synergids and the two polar nuclei will be there. Two polar nuclei comes in what? The central cell. Now this is the presentation of insect pollination. So if you are asked what is happening up over here, this is insect pollination that is happening up over here. Now this is a plant where you get two type of flowers, clistogamous and chasmogamous. Clistogamous, as in the flowers are closed, they would be having the androecium as well as gynoecium together. So there will be obviously pollination happening, and this will be always autogamy. So there will be seed set formation for sure in clistogamous flowers, that is the closed ones. In the open flowers, there can be gytonogamy, there can be xenogamy, there can be autogamy, whatever you want, there can be there. Now, what are these flowers? Viola, oxalis and commelina, which would be having both clistogamous as well as chasmogamous flowers. Cool? Going ahead, typical dicot plant structure, but more importantly you have LS of embryo of grass, that is monocot, my dear children. This is very important picture. So monocot have just one cotyledon. The name of that cotyledon is scutellum, my dear children. They also have plumule, which is shoot apex. It is covered by something that is known as coleoptile. They also have radicle, that is root apex, and is covered by root cap and coleorhiza. So all this structure is really important. You should be knowing to diagram. This is a very important diagram, just like the antibody structure. Okay, now this can be given for identification. You can just go through it. Structure of monocot seed, the exact thing that is put over here. So coleoptile, plumule and radicle down, coleorhiza there, scutellum towards one side, and endosperm is also persisting. So there will be having some amount of endosperm still left, so that is being mentioned over there. Okay, children, do not worry about all these pictures that I am showing you over here. Is this something to worry? No, you're getting the ready-made resources, so why do you have to worry? You just have to practice, and we can practice now. We have still days left to practice, so please practice it in the best possible way. Okay? Now, one more very important thing. So if you're someone who is a KCET aspirant as well, so we have Abhyas KCET test series only for you guys. So I know PU2 phase might be going on as of now, so it's okay. You can take your time, just see through it. If at all it works for you, there is free part test that is available. You can go through it as well whenever you have time, and then you can enroll to the Abhyas KCET test series, where there will be 35 full syllabus and chapter wise tests. There will be video solution, there will be one shot revision videos, okay, and also 5-year paper analysis. That's all for ₹1499. And as you are in PU2, you can also avail the PU2 test series, which will be available for just ₹1. So it is actually 1498, but for ₹1 you get it extra. So prepare, practice and perform. Going ahead with human reproduction, my dear children. Over here you have diagrammatic view of male reproductive system, and then you have a sectional view of the female reproductive system. This is very, very, very important. Okay, so over here please know the testis, primary sex organ, then you have accessory ducts carrying the sperm, then you have accessory glands providing secretion to the sperm, then you have external genital organs. Similarly over here you have the you have the ovary which produces the egg, and then you have fallopian tube which takes it. Uterus is also a part of accessory duct, and vagina is also part of accessory duct. Okay, then that's all. So at least this structure is really, really important. Go through it clearly. Okay. Now one more picture is there which is also important. Again you might feel ma'am is saying all important diagrams. So antibody structure, the sectional view of this thing, what do you call it, anther, and then we studied this particular, okay, then there is monocot embryo, then you have sectional view of reproductive system, female reproductive system, and then this thing is also very important. It's there in the model question paper as well. So you should be knowing that we have almost 15 to 20 mammary lobes. Inside the lobes, my dear children, this small things that you see, they are alveoli. So alveoli open into tubules, and tubules are not being shown up here. Suppose this and this is a tubule. Tubules will be opening into duct. Duct will be opening into wider region, that is ampulla. Ampulla will be opening into lactiferous duct. That is how things work. And then there is some fat which you have to put here and here. This is nipple, this is areola, and these are the ribs, and in between ribs you have the muscles as well. Cool. And then this is a schematic representation of spermatogenesis and oogenesis, which might also be asked, but if you know the concept you would be able to answer it really, really well. So spermatogenesis and oogenesis starts with spermatogonia and oogonia, which undergoes mitosis, differentiates, and then you have primary spermatocyte, there you have primary oocyte. Both of them will undergo their first meiosis, form secondary spermatocyte over here. There only one secondary oocyte will be formed. There will be one first polar body that will be degenerated. Okay. Now both of these secondary spermatocyte will be giving rise to spermatids. Now spermatids will undergo differentiation again to form spermatozoa. This process is known as spermiogenesis. Now over there the secondary oocyte will be dividing again into one ovum and second polar body. This will again degenerate. So from one oogonia basically you are, or basically from one primary spermatocyte you are getting 4 sperms, but you are getting only one ovum here. So just know that difference, and also know what is haploid, what is diploid. So primary spermatocyte and primary oocyte is always diploid. They are going to start meiosis. After that they will be haploid in the next stage. Remember it. Okay. Human reproduction, diagrammatic representation of the menstrual cycle. This picture can be given, you would be asked, where is estrogen and progesterone released from? It's the ovary. Where is FSH and LH released from? It is the pituitary gland. What is the role of the hormones up here to maintain the endometrium, to do things like that and thing? Okay. Now over here the embryo formation you should be knowing. Morula is the 8 to 16 cell stage, and in which stage does it go and get implanted? It gets implanted in blastocyst stage, my dear children, not any other stages. So morula means 8 to 16 cell stage, then it will roll down, divide and form blastocyst, where you would be where it will be finally getting implanted up over here. Now evolution chapter, you have S.L. Miller experiment, my dear children. This is very much important. Over here in this chapter there is only one picture that you should be definitely knowing. So boiling water, the water vapor comes in, you have electricity, that is light basically, to provide temperature. It's not light, it's basically to provide high temperature, that is 800 degrees Celsius. Now at this high temperature and also reducing environment, there is no oxygen, there is only CH4, that is methane, ammonia, H2O and hydrogen. In this condition what will happen, my dear children? Certain organic compounds would be formed over here, such as amino acids and stuff like that. Okay, so this is the S.L. Miller experiment. Just go through this picture really well. Now, reproductive health, you have two pictures just to know, okay. So, this is vasectomy where the vas difference are being cut, tubectomy where the fallopian tube has been cut and tied. These are surgical methods of contraception. Now, these are implants. If you are asked what is this thing, you should be knowing this is a contraceptive method, that is implants which is put in the skin and they will be releasing hormones, okay, such as fluctuating the hormonal level in the body, avoiding ovulation and fertilization to happen. Okay. Now, microbes in human welfare. There's a picture of the biogas plant on page number 156. So, this is where you add the dung, you mix it with water and this goes over here. Now, this is where there is no oxygen. So, the anaerobic bacteria will be giving rise to biogas and the biogas will be moved from the outlet. Whatever thing is left behind, they will be going to this, okay, and here they can be utilized as a fertilizer or manure. From here it can be taken for getting used as a fertilizer or a manure. That is about microbes in human welfare. Now you might be thinking that okay, it is getting much easy. I'm telling you it will be easy. Now you have biotechnology principles and processes, my dear children. Now this picture can be given, you would be asked about sticky ends, okay, how are they produced, what is their use and things like that. So sticky ends are very much important in recombinant DNA technology because you are going to use the same restriction enzyme to cut both the vector DNA as well as the plasmid DNA and the nucleotide, basically the plasmid DNA and also the foreign DNA that you want to insert. So the sticky ends, if they are there, they will obviously find their complementary base pair and just bind with it. Okay, apart from that, you have PBr322, that is a plasmid. All the sites over here are really important. So this is the origin of replication site. This is ROP. This is protein is really important for DNA polymerase to come and act. And here this is tetracycline resistant site. You have a site for two enzymes, BAMH1 and SAL1. Okay, this is ampicillin resistant site. Again, you have two sites for two enzymes, that is PVU1 and PST1. Now, PVU2 is an ROP site, my dear children, and over here you have EcoR1 clavin site and HIND3 site. So all this enzyme site you should be remembering, and also the resistant gene that is there you should be remembering. A very important question. Okay, going ahead. So this is again something that is very important. Biotechnology principles and processes. Bioreactor is there. This is a stir tank bioreactor. So what is the use of it? Basically the air will be spread through evenly. That is the use of it. Okay, and over here you have polymerase chain reaction. The steps would be asked. Denaturation is first thing, then is annealing, then is extension. So you could be asked what is involved over here. So it's DNA polymerase plus what? DNA polymerase plus DNTPs, that is deoxynucleotide triphosphates. So that means nucleotides. Okay, they are in the form of triphosphates. So each nucleotide that you see, my dear children, they are come somewhat like this. 1, 2nd, 3rd, 4th and 5th carbon is here in the pentose sugar, right? Now this carbon is attached to phosphate group. This is what we learned, and this is attached to nitrogen base. So actually it is attached to 3 phosphates, and when you want to pair it with some other nucleotide, this bond is broken down. So it is initially deoxynucleotide triphosphate, 3-phosphate, but when you're breaking it down, this bond's energy, okay, which is being stored in the bond, is being provided to make bond with the other nucleotide which is there. So deoxynucleotides and also DNA polymerase is being used up over here. Cool? Now, biotechnology and its application, you had the structure of insulin, my dear children. You have A peptide and you have basically this is A peptide, this will be B peptide and this is C peptide. So this is proinsulin because it has a structure like that. Now in actual insulin, my dear children, which is the functional form, you remove the C peptide. So A and B are together, you're removing the free C peptide, that's when you join it down, that's when you get insulin. Now what are the bonds present over here? These are the disulfide bonds present between the chains of the polypeptide of insulin. Okay? Chalo. So that is important. Now organism and population, over here you have logarithmic and exponential graph. Exponential as in there is nothing that is limiting, everything is increasing, the population is just growing. Logarithmic is when you have limited resources, you're considering that there is no abundant amount of food, there is no infinite food, there is no infinite water. So in the limiting resources, the growth will be like this. This is known as logistic growth. This will give rise to a sigmoid curve, this will be giving rise to a J-shaped curve. This is the formula dn by dt is equals to Rn for exponential, and for logistic it is logarithmic, it is what? dn by dt is equals to Rn, k minus n divided by k. So what is k? Carrying capacity. That means that's the only limit that I can carry up to. This particular population can be carried up to. Okay? Yes. Next thing, ecosystem, my dear children. These pyramids which are there are really important. Pyramid of number in grassland ecosystem, you would be asked to make one, okay, with examples. Then pyramid of biomass. Please remember, in the case of aquatic ecosystem, I think I have drawn it, shown it, yeah. In the case of aquatic ecosystem, the pyramid of biomass is inverted. Why? The phytoplanktons which are there, they have little biomass, and the animals which feed on it obviously would be having more biomass than it, which is why it is an inverted pyramid in the case of aquatic ecosystem, biomass, okay. Now if you talk about energy pyramid is always upright, never inverted. It is always upright. Cool? Okay, so if you're worried not about just biology but about other things, so here is a detailed schedule, my dear children. You can just go through it. This is for chemistry. If you want, you can take a screenshot, okay. Now going ahead, I have for math as well. This is all for you guys, so you should know when is the video coming. You can just be prepared for that, right? You can allocate your time accordingly. Now for physics, you have these many days. Fine? Chalo. Now how to remember diagrams. So with this we are completing all the diagrams, my dear children. But there's a question, how do I remember the diagrams, ma'am? I cannot remember it. So the first thing that I will suggest you to do is, okay, little amount of animation. Understand the diagram, understand the concept behind it. You would be able to remember the diagram for a longer, and also the labelings. Now the first trial is when you are going to see and draw. You look at the picture and you draw it as such. Now, the second trial is when you are going to draw but without reference. Okay, so close your book and you should be drawing without reference. Now one very important thing that you have to do is you do not have to label this. Oh sorry, I meant basically you are not drawing without the reference, right? So you drew it. Now when you open your book and relabel it, you will be finding that I have not made this thing. How can I label it? So what will you do? You will again make it fine. That is a mistake that you did, but you rectified it. In that way you would remember. Okay, so in that way you are practicing labeling also. Now one more time you will draw finally, and you will keep it unlabeled for some time. You would be labeling it after some time. In that way you are basically helping your recall power go better. So you can do it now and keep the labeling for the time during the exam, like a day before the exam. You have 2 day gap, right? So the first day you can try doing it. So draw it now and keep it unlabeled. So understand the drawing really well, keep it unlabeled, label on the day of, just before the day of exam, not on the day of exam. Okay, so that's how you can remember the diagrams better. There is no other shortcut to it. So that's a problem. If I find some, I will tell you. Okay, so that's all, my dear children. Okay, my animations never end. So I hope that this particular video was helpful for you. So you have to tell me if this was helpful or not in the comment section, and also do let me know if you have any other queries that I can solve. Okay, and lots of love. God bless you all. Bye bye. Take care.